So, hello everyone. I am Gyanjoti Raskwa, convener of this webinar. Welcome you all. Hope all of you are staying safe, healthy at home. Now, I would like to invite the faculty of our department, Dr. Lokhi Burhagohai, to deliver welcome speech. Dr. Lokhi Burhagohai, over to you.
Okay. Good morning to everyone, and thank you all for joining with us for today's webinar. This is the first initiative of our department to organize such an event in the time of pandemic using the new technology of communication. On behalf of Physics Department, Soidwar College, first of all, I would like to welcome Honorable Resource Person of today's webinar, Dr. Paramananda Soikia, for joining with us. Dr. Soikia is a senior scientist, ANDE, Greater Boston, USA. Dr. Soikia is a molecular biologist having more than 15 years of research experience. Next, I would like to welcome our respected principal and inaugurator of today's webinar, Dr. A.K. Uza sir. Due to his constant inspiration and support, we are able to arrange this webinar. I would like to welcome all the respected participants from different colleges as well as our host college, research scholars of different universities, and friends. Our students are also participating in this webinar. I extend my heartiest welcome to them also. The theme of the webinar, Biophysics and Medicine, is very relevant to the present day situation. I hope the webinar will be an informative one and everyone will be benefited. Thank you.
Thank you, Dr. Lokhi Burhagohai. Now, I would like to request our respected principal, sir, Dr. Anjan Kumar Uza, to deliver his inaugural speech. So over to Dr. Uza, sir.
Thank you, Gyanjoti. Good morning, everyone present here in this international webinar. Department of Physics, Soidwar College has organized this international webinar, and I, on behalf of Soidwar College fraternity, extend my heartiest welcome to all the participants, scholars, students, and teachers. Dr. Paramananda Soikia, senior scientist at ANDE, Boston, USA, is joining with us as resource person. The topic of today's seminar is biophysics and medicine. Biophysics is a field that applies the theory and methods of physics to understand how biological systems. Biophysics has been critical to understanding how different parts of a cell move and function, and how complex systems in our bodies, the brain, circulation, immune system, and others work. Biophysics is a vibrant scientific field where scientists from many fields, including mathematics, chemistry, physics, engineering, pharmacology, and material sciences, use their skills to explore and develop new tools for understanding how biology, all life, works. Physical scientists use mathematics to explain what happens in nature. Life scientists want to understand how biological systems work. These systems include molecules, cells, organisms, and ecosystems that are very complex. Biological research in the 21st century involves experiments that produce huge amounts of data. How can uh, biologists even uh, begin to understand this data or predict how these systems might work? This is where biophysics come in. Biophysics, Biophysicists are uniquely trained in the quantitative sciences of physics, mathematics, and chemistry, and they are able to tackle a wide array of topics ranging from how nerve cells communicate to how plant cells capture light and transform it into energy, to how changes in the DNA of healthy cells can trigger their transformation into cancer cells, to so many other biological problems. Biophysicists work to develop methods to overcome disease, eradicate global hunger, produce renewable energy sources, design cutting edge technologies, and solve countless scientific mysteries. In short, biophysicists are at the forefront of solving age-old human problems as well as problems of the future. The topic is really a very emerging one, and we are inviting Dr. Paramananda Choikia, famous molecular biologist, to deliver his speech on this topic. Last year, Dr. Choikia uh, came on our invitation to Soidura College to address an induction program for our students of the college. He encouraged our students a lot with
এটা
his
রিমুভ নকৰে দাও
celebration and interaction
এজন আমাৰ ষ্টুডেণ্ট
with them. This time also, he has accepted our invitation in spite of his busy schedule. Really, we are very grateful to Dr. Soikia. I wish that Dr. Soikia's deliberation would open some new aspects and would be able to enrich us in the new field of knowledge. Wishing a great success of today's webinar, I declare that today's webinar is being inaugurated. Thank you all.
Thank you, Principal Sir. Your inaugural speech was so informative and beautiful. Thanks a lot. Now it's time to introduce our most awaited research persons, Dr. Paramananda Saikia, in front of you. I take this opportunity to introduce him in front of you. Really, today I am very much honored and privileged to introduce Dr. Paramananda Saikia in front of you. A journey which started at Thekiajoli High School today has reached at Boston, USA, is not so easy to describe in front of you within few minutes. Yes, I really mean about Dr. Paramananda Saikia. He born at Tezpur, started his academic life at Thekiajoli High School. He went to Darong College, from where he got his graduations. He received his master degree from Tezpur University. He had been awarded with doctorate degree from prestigious Indian Institute of Science, Bengaluru. He then went to USA for postdoctoral research in the year 2005. From 2005 to 2011, he is at Cleveland State, Ohio in USA. In 2011, he came back to India and started his service life as an assistant professor in the Department of Biotechnology, University of Hyderabad. During that period, he guided many research scholars, published many international papers, but his interest in research and his curious mind brought him back to the USA, where he started his career as a research scientist at Cleveland Clinic, Ohio. And today he has become famous as a senior scientist at ANDA, which is one of the global leader in DNA technology. And today we are happy and fortunate to have such a person with us as a resource person. I'm sure his speech on the topic will enlighten us. With this, I would like to invite Dr. Paramananda Saikya to deliver his speech on the topic. So over to Dr. Paramananda Saikya. Dr. Saikya.
Hi, yeah.
Okay.
Good morning. Yeah, good morning, everyone. Can you hear me?
Yes.
Yes.
You're audible.
So I'm okay. Okay. So yeah, that's good. So yeah, first thing I would like to apologize for the technical difficulties from my side. As you know, now everybody here also so things are happening slowly, getting better, but still we're struggling in some part in this technology. In sometime we get good connection, sometime we get bad connection. So, but we're doing better. So, with that, I would like to give some kind of introduction. But first, I would like to thank our regarded Professor Dr. Rosa. He gave a very good introduction about biophysics, and I thank him for giving me this opportunity to explain my knowledge about biophysics and how we can use this biophysics in healthcare mostly. Then I'd like to thank my friend Dr. Gyanjoti for he also giving me this opportunity to come and talk in front of you, meaning like sharing my experience on biophysics. So with that, I'd like to tell a little bit about my background, but already Dr. Rosa told me about what I have done and all. But currently, I'm a senior scientist in a DNA diagnostic company in Boston.
Yeah.
So what we do in our company is that if I siblings, if somebody is related to somebody and we don't know the identity, then that's called kinship analysis, basically, that we identify people basically based on genomics. So we do a lot of work, but our main field is genomics. So until I joined this company, I did not realize how powerful our genome is. Because all of you know that we all human, not only human, all organisms on Earth have DNA as a genetic material. So, and the genetic material is not very different from each other, one person to another, within a family or within a population or in a whole world. We're very much related to each other. Saying that we're almost 99 individual people, right? Because we have only 0.1% different. So in our company, what we do, we use that 0.1% chance, how they're different. and within the difference, there's some kind of identity between same family member or brother or sister. So we use those 0.1% sons and identify people. So it's a kind of a challenging job because we have to study in a population. Population is a little bit bigger, so it's a kind of science, but still, it's very successful technique basically to identify people. Second thing is that we are now all of are going through a very harsh time. It's called COVID-19 pandemic. So we are all affected by that. And another problem is that in COVID-19, we have to have a system to identify. That identify means like the people that is infected. And what can be done to identify is that we need some kind of tool to identify, right? So, and there are so many people, and some people are symptomatic and some people are not symptomatic. Symptomatic means like, they will show you that, their person is infected with the virus and having some kind of symptom. But sometimes, like, people also get infected and they don't show any symptom. Then how will you know that, these people are infected or not? So it's a kind of very challenging because sources are limited. How many people we can test? If we have a 1 million people and we don't have that much, I'm just giving a number, but we have suppose 1 million people to test, but we don't have a 1 million, like, we have 50,000 kit to test. So our company, like, once this pandemic start in March 2019, as a genetic company, we jump on into it, and we almost develop a kit to identify. Identify means like, a detection kit. So based on genomics. So we're kind of a genetic company, genomics company, and doing a lot of studies like that. So with saying that, I'd like to share some kind of some slide. I don't know to uh, put a lot of data and explain those data, and because, like, that I don't feel that, showing too many things, but it's more complicated. But better give some concept, or give some kind of idea that how this biophysics can be used in our daily life. Daily life means like, we need uh, like tools or a kind of technique to study stuff. So I'll give you some kind of idea about that, and I will also tell a little bit about the scope. Scope means career. How we can use biophysics, taking as a subject what we can do in our life basically. So, and I would like to keep this presentation as a very informal, like, kind of a discussion type, not a kind of a course. So, but I'll be available after the this presentation as well. If you have some doubt or you want to learn something more, I'll be very happy to help you or guide you, or provide you the information. And I can share my email address to the organizer, so that you can take from them and you can contact me. So with saying that, I'll share some slide. Sorry, it's kind of.
Proceed. Go.
Give me 1 second. I'm not sure where the files are.
Screen share কৰিব পাৰিবতো কো হচ্ছে
Give me 1 second. I'm not sure why it's not coming.
কি হলো
Can you see the slide? No, right?
No, it's not coming.
Wait. Uh,
কি কৰি আছে?
come.
ৰবা ৰবা।
Mm, okay. Okay. Yeah. So yeah, now you can see it,
Okay,
right?
okay. It
Okay.
will come. Yes, we can
Okay.
see your slides from. Yes.
Yeah. Okay. Good. So now let's go to the next slide. So let's talk about a little bit history. I don't like that much history, but but still we should remember how things got advanced and how things got started. So as Dr. Oza sir said already that what is biophysics is nothing like it's an interdisciplinary science. It involves physics, biology, chemistry, and mathematics. So it's a kind of systemic study of a particular biological problem. Now, if you see, like in the slide, 1892, Carl Parson defined this as a like missing link between biology and physics called biophysics. And in 1946, like biophysics research unit, King's College, London, they hire like physicists who work on questions on biological problem basically. And maybe all of you know about Maurice Wilkins, Rosalind Franklin. So they did X-ray diffraction of DNA. It's a big things in molecular biology, like the three-dimensional structure of DNA. So how DNA is for, like how what is the DNA structure? They did X-ray crystallography, and we know how DNA look like basically. So then in 1953, Francis Crick and Watson basically proposed the DNA double helix structure. So why I'm saying about the DNA double helix, like this is the main component in the biology to understand a lot of stuff. So in 1957, the Biophysical Society founded. So it's a big society. So and who take care of like biophysical scientific development, any kind of up to date research. They keep everything in their system. So you can see that how things actually from 1892 to 1957. So it's a long journey to get well recognition of this branch of science. But saying that, it's not that people have not done biophysics research before that. People has done, and biologists always used physics tool, physics concept to understand the problem of biology. So if I show you this cartoon, it's better explain what is a systemic studies of a biological problem. Not only biological problem, but other problem as well. So if I am like a kind of a pure biologist, so you can see in the top part, like the person sitting down. So as a biologist, I'll think that it's a fan, like touching the ear of elephant basically. But as a other, like as a chemist, like I'll feel that it's a sphere, like touching the tusk of the elephant. Or like a person like sitting the trunk, it will feel like a snake. And like environmentalist will feel like it's a tree touching the feet of the elephant. And the scientist in other branch will feel like it's a rope touching the tail of the elephant. So the picture says that until unless we study the all part aspect of the problem, we'll not understand that it's elephant. So you can understand the the concept behind this. So now we in a biophysics approach, like we integrate all the branch of the science, like as like mathematics, physics, chemistry. So to get a holistic view of the problem. So these pictures, I like it, and it's very easy to explain, right? So as a biophysicist, what we generally do, or what's your goal in biophysics? So first thing what we do, we create simplified model of biological system. So what is a biological problem? Like, it's not easy to say the biological problem, or like, no, I'm not talking about problem. Problem is like what we don't know so far. We want to understand why it is happening, how it is happening, right? So in biophysics, like, we try to simplify the model. Second thing is that what we do is make quantitative prediction. Like, see, we cannot do everything, right? In the laboratory, like, we have limitations. So we have to have a quantitative prediction, like, what can happen to minimize our laboratory work. Then if we see that, in quantitative prediction that is happening, I'll tell you with some example what do you mean by quantitative predictions. If we know, like, okay, we basically narrow down the whole system, then we can experimentally test quantitative prediction. So that means we come from a big picture and narrow it down, and but ultimately we solve the problem basically. So um, if we say, like, the biophysics, what we can do in biophysics, like, we in biophysics you can do like molecular and subcellular biophysics. Like, these are the branches I'm talking about. They are not the fixed branch of biophysics, but there will be a lot to. Okay, the structure and conformation of biological molecule. Okay, their functional relationship. I'll go, like, slowly one by one. And conformational transition, ligand binding and intermolecular binding, diffusion and molecular transport, membrane biophysics, DNA and nucleic acid biophysics, protein biophysics, energy flow and bioenergetics, and thermodynamics. My whole idea of putting this slide is that to appreciate that, as a physicist, there are so many, area to do research, or there are a lot of things to understand. So you can see that, there are so many things in biology, so where, like, we can apply our physics to understand. Then, okay, now let go a little bit deep into the biology a little bit, but I don't want to give you a lot of like, complicated things of biology. I know many of you may be from physics background, mathematics background, or chemistry. So but to say biophysics, I think we should little bit know about these four molecules that we found in living organism. So they're called, like, macromolecules. One is called DNA, deoxyribonucleic acid. Then second is called protein, and third is called a lipid molecule, and fourth is carbohydrate. So I think, although you are from maybe pure science, I think all of you know DNA, protein, lipid, carbohydrate, right? So to make it more sense, like, actually I train as a virologist in my PhD, and I have done like, 10 years, more than 10 years of research in virus. But I was always like, worried to talk about virus, like, to the common people, because I feel that, they will not understand, or they will have kind of inhibition to know more, or something like that. But now, after this COVID-19 pandemic, I think every person on earth knows what is virus. So I feel proud of it. I like, in a sense that, everybody know what virus is now, and how important it is to do research on virus. Okay, now coming back. So DNA, deoxyribonucleic acid, how you can see that, they're a linear structure. Like, you can see how they form. I'm putting a B form. Like, there are different conformation of this DNA molecule. So there's a form called B, which is commonly found, which is like, energetically possible, basically most commonly found. So then protein, like, I'm putting an example of hemoglobin. All of you know that, hemoglobin, what it works. It carries oxygen. And lipid molecule, there's an example phosphatidylcholine, and there's an example of carbohydrate, like branch complex carbohydrate. So these are the four main component we found in a living organism inside a cell. Now, if we say, like, now I'll go a little bit in, but I'll not go very detailed. So if you say, like, nucleic acid, it's a kind of polymer of deoxyribonucleic acid or ribonucleic acid. There are 4 different types nucleotides, G, T, C, A. Then protein made of amino acid, all of we know. So basically what happened, in a living organism, there is a kind of continuation of these nucleotides or continuation of these amino acids to form a particular structure like protein. If we say made up of amino acids, and DNA or RNA is made up of nucleotide, but ultimately it is environmental component that determine how this molecule will be formed in 3 dimension. Like you can say these are not linear structure in real life, so they will form a different structure basically. So why it is important now, DNA and protein. So what happened, in any organism, our genetic information is stored in nucleic acid, right? Either in DNA basically. But I should mention here that DNA is a genetic material, but RNA is also a genetic material in many organisms, like COVID-19, that is the RNA virus. So their genetic material is ribonucleic acid. So there's some chemical difference between these 2 molecules, but I don't want to go in detail that. But we know we can discuss now. Our genetic information is stored in this nucleic acid, right? So now what happened, sometime when organism grows, or people grow, basically the cell multiply, right? So this nucleic acid has to be multiplied too, but it happens sometime there's some error happen, it is not always perfect. So some kind of mutation, I think all of you know some this word called mutation. So mutation is nothing that it got changed basically, which is not in real form. So because of that mutation, people get disease. So why it happened? Because the genetic information flows from DNA to RNA, RNA to protein, and protein is the action molecule basically. So if something mistake happen in this nucleic acid, ultimately the protein that is made in the cell is not properly folded. Folded means it's not getting their structure. So you can just say example, like suppose our hand, if our 5 fingers are we have 5 fingers, like suppose we have if 2 fingers connected or 3 fingers connected, and we cannot do our function properly, right? So but if you look at mm, uh, in developmental biology, these fingers are getting separated, so that we can do proper function. So like that also protein, when they synthesize in the cell, they have to go through a proper kind of a maturation, folding, so that they can do the function. So now okay, so now let us go to the problem, let us discuss. So now as a biophysicist, so what we can do, suppose as I was talking about protein, right? Protein is synthesized in the cell, and they synthesize, then they fold, then they do function. Now how we'll study how this protein look like, because it has a kind of a 3 dimensional structure. So as a biophysicist, what I'll do, as a I'll do the extract crystallography. This is a technique called extract crystallography. All of we know that it's a kind of diffraction of the protein. It's a biomolecule, right? So we first what do we do? We purify the protein from the microorganism, or we have a system to purify. Then we purify the protein, then we crystallize. So there are different kind of medium that we use to crystallize this protein. Once the protein is crystallized, then we do X-ray diffraction. So basically with the diffraction, we can determine the structure of the protein. So it is not very easy to crystallize a protein. If any of are you in the audience know about biophysics, crystallography, protein crystallography, you understand that it's very hard to crystallize the protein, because because we don't know the exact atmosphere inside a cell that needed to protein to be crystallized. So but there are a lot of experimental protocol that we test, and people were successful in getting a lot of protein 3 dimensional structures. So as a biophysicist, like we purify the protein, then we crystallize and do the diffraction, and then we start analyzing the protein structure. So now, why we need to know the protein structure? Because many of, like not many, all our body metabolism or any body chemistry happen, it needed enzymes. All of we know, right? We need enzymes in our body. So these enzymes, or nothing, is a kind of protein. So now these enzymes uh, to always act on a substrate, right? Always enzyme act on substrate. So like as a carbohydrate, if we eat carbohydrate in our food, like that has to be digested, right, to make a simple sugar so that our body can absorb. So like this, like our protein basically have to bind to our substrate in a perfect manner, meaning they have a active site, like where like they have a very definitive structures where like our substrate go and bind so that they can do the chemical reaction to digest or like break down. Now if there's some kind of mutation in the protein gene, basically gene, then ultimately the protein will be not folded properly, like suppose it has to form like this, then it will be like it's in a closed structure. I'm not so sure whether you're seeing my finger, but supposed to be open, but they are closed in one direction, so our substrate cannot get in. So uh, we by protein's three-dimensional structure, we can see basically that how things got changed, and we understand like what is the problem. Now second thing is that in pharmaceutical science, we always I didn't discover like drugs, right? Different kind of drugs we saw in the market, right? So they are not nothing but like they are like competitor for a like in a particular substrate, for a particular enzyme. Or what happened, like in a particular protein, like they have a different site, like activator site, or like different site of the protein, where they, if something go and bind, their structure gets changed basically. So they cannot bind to the substrate basically. So that means we synthesize or we identify a lot of small molecule, like molecules, compounds that we test in the laboratory, whether they can bind to the particular protein and they can inhibit the function of the protein. So why I'm saying is that sometimes we get infection, right, by virus or bacteria or other like um, other microorganism. So then when they come inside our body, like they generally synthesize their own stuff, right? They were having their enzymes, then they multiply, then they grow. So what we do generally, like we like study a lot of small molecule that can go and bind to the viral proteins, like specifically the viral proteins, and they bind and inhibit the function of this particular protein of the virus. So now you have to be very critical, like we should not develop something like that goes and bind to the human protein, right? Because if we inject some drug or like in a compound that go and bind and in human enzymes and it block it, then our regular like physiological function will be not done. So it has to be very specific. So now the biophysics comes over there too. Like we study the drug protein interaction, so how they react basically, and we have to be very specific, like designing the small molecule, how they go and bind. The other problem is that as I said, like it's not very easy to crystallize all the protein and study. Now there's a called computational biology. What uh, we do a modeling. Modeling means we study like similar protein in like in a lot of database that we get the information, and we just model based on the amino acid sequence. So that kind of studies is very helpful because it's very less expensive, because we don't do the experiment really, but we do in a computer the modeling. So we do a lot of things like that. Now comes to like current pandemic problem, like the COVID-19. So let us talk about a little bit about that virus now, how we can do something applying biophysics. So all of you know that there are two kind of treatment to a particular virus infection or like bacterial infection is that one is preventative. So where we like inject a particular human with a component of the bacteria or virus, then they generate antibody in the body in the human. Then when there will be infection, like they already the body will be prepared, so there will be no infection. But there may be infection, but it will be not multiplied basically. So that is called preventative. Then another is called therapeutics. Like already the infection happened, and we have some drugs we put it, and drugs, as I said, like they can go and inhibit some particular enzymes, or they break the cell wall of bacteria so that they can multiply. So these are the two things that we do in medicine biology. So now coming back to vaccine, vaccine is a preventative treatment. So now you think about it, like how vaccines are developed. As I said, vaccines are nothing, it's a part of infectious organism, either like virus or bacteria. It's a we sometimes people take the whole virus or whole bacteria, they inactivate it, then they inject it basically. Or sometimes what they do, they take like part of the bacteria or part of the virus basically, then they inject to the human, then they generate the immune response to the body, and they protect basically. Now the big question is that virus is also having a lot of stuff, right? It has a particular membrane, it has a lot of nucleic acid, it has protein, different kind of proteins. Now and now the problem is that it's a new virus, we don't know anything, not a lot of stuff. So what we have to do first, that we have to look for some protein of the virus that highly exposed. Exposed means they are on a surface of the virus. I'm talking about COVID-19. So some particular part of some proteins of the virus that is outside of the virus. Outside means they are basically exposed, like hydrophobic. They'll be now, and we know this genomic sequence of the virus. And if we know the genome, we can predict the protein sequence as well. So now if we know the protein amino acid sequence, then we can predict what particular part of this protein is more hydrophobic. So by studying that, we can use that particular part of the protein for immunization. Immunization is nothing, is the kind of we inject to the human to get immune response. So now you can see that it's a lot of stuff. Bioinformatics analyzing the sequence of the protein, then predicting the most hydrophobic part of the protein of the virus. Then ultimately we inject that part, right, and we get immunity. But see, another thing is that this is a kind of a prediction, right? We have not done the experiment yet. So and we don't have that much time because the virus came so fast, and we do not have any drugs, right? So we have to rely on this bioinformatics. So it has been done that way, people has predicted, and people already almost 75% work is done to the vaccine development. So now you see the power of the biophysics. Not only biophysics, as I say, it's a kind of integration of different science, right? Computational biology, the biology immunologist, or physicists, all of them come together and do research to solve the problem. Now there are a lot of things like that, where you can we can solve the problem using the physical properties and physical method to understand the problem. So saying that, now if I come to the uh, most important part, that why should I study biophysics? What is a career? What I can do? So there are a lot of things you can do. First thing is that as a biologist, I'm a biologist, so I feel that if a physicist come, they if they understand the chemistry of this biomolecule, protein, DNA, carbohydrate, lipid, and they can do a lot of good research. A lot of good research means that uh, like to understand the detail the structure of this biomolecule, then they can understand like in disease condition like how these biomolecules are formed, and there are a lot of method that we can use to study these. And as a physicist, like it will be easy for them to understand like the basic mathematics or like that kind of related stuff. So because like when I'm a biologist, I'm solving a problem. My always thought is like to understand deep into the biology. So if a physicist come like they can integrate, and they can with a combination you can do a lot of stuff. So people can like anybody can take career in biophysics in research. Uh, then they can go in academia like the teaching or like doing research. And second thing is like it's a very high demand in industry, in pharmaceutical company. There I think you may be knowing like Genentech, uh, Johnson and Johnson, uh, this big company like they Novartis. I think all of you heard about this company. They're AstraZeneca. They all look for like biophysicists, because as I say like they're behind developing drugs for a lot of diseases. We still don't have a vaccine for HIV. We still struggling for mycobacterium tuberculosis. Still we don't have it. Zika virus. There are a lot of uh, diseases like still we don't have any cure, or still we are in a struggle. So if like biophysicists come to a company, so they can study all these drug protein interactions, and they can do like structural studies, because structure is very important for function, right? So it's a good branch of science. It's emerging, uh, to because as I say, like I saw the picture, like in the beginning, that the elephant picture, like so if a physicist and biologist or chemist and mathematician work together, so we can solve the big problem. So early days, like in 90s, like these science like biologists, what they do, like they do study like going A to B to C to D, like it's very linear science. A lot of people were doing, and like people from pure science, like they did not appreciate that much biology, because they're like whoever like not doing that much science, they go to biology, like they just do some experiment, they do flower, like like botany, zoology, that kind of study people used to do, right? Earlier, I think before Watson Crick double helix structure, like people just simply doing some kind of experiment. So but after 1970, like after DNA double helix came, a lot of interdisciplinary science emerged. Another thing I'm telling about system biology, which is more towards like it's a holistic science, mostly mathematician and physicists. Basically, they come and do some modeling, basically, because as I say, like earlier people used to do study like how A become B, become C, C become D. Now there's another thing like X, Y, Z. 1, 2, 3, they all are basically interlinked, not like that way. So if we study all like system wise, then we can understand the problem. So uh, they are very appreciated, the biophysicists in the industry as well. So there are a lot of scope, not that not in academics, but in industry as well, like with a good job. So I think I'm good in that, and I leave like more time towards any question and answer or any kind of discussion. If we have anything, like it will be more. Um, I think it's better than like giving more slides or data. So Gyanjoti, anybody like want to have any discussion or any question? I'll be happy to do that.
Hello,
Yeah.
respected participants. Respected participants, if you have any questions, any doubts, queries, you can directly ask to our resource person. He is ready to answer. You can directly ask to our resource person.
I think you have to unmute, right?
Participant can unmute themselves.
Hello, sir.
Yes.
Hello,
Yeah.
sir, and Ritu Sekia, sir. Sir, I am Pubanita Bhuya. Sir, I have two questions. So,
Sure.
first question is, what is US's status regarding COVID-19 and vaccine? What is
Okay.
US's
Yeah.
present status?
Actively engaged in COVID-19 research, and I'm in Boston, and the company maybe heard Moderna. They're trying to develop the vaccine using mRNA. mRNA is like messenger RNA based vaccine. So what they did, like they have designed the mRNA, then they vaccinated basically with a small population, I think 35 people. Then they got a very good immune response in them. Then they went ahead and did, I think, 30,000, 25 to 30,000 people immunization, and they are now in the verge of checking their immune response. So by November 2nd, they're expecting that the vaccine will be in the market. Like I'm talking about USA status, not like I heard that in Russia they already immunized somebody, and Oxford also they are saying that they have the vaccine already. But in US, like that I can say that they are in the final stage of checking, because not only that we should have a vaccine, but it has to be safe, because there are a lot of cases, like about dengue virus, right? Dengue virus. So in Europe, in Spain, basically they have developed a vaccine. I think some years back, they immunized, and a lot of people died basically because as I said, like these are made with live virus, or something like that. So there is a risk of safety. So in US, like they are more concerned on that. So they're still doing, but we're expecting that in November, I believe, that will be there.
So my second question is, I am currently doing PhD chemistry. I don't have any connection with biology. Can I ever join in biophysics research in USA? And if I can, how should I proceed?
Correct. Yeah. So if you see, like all the, like successful scientists, successful means like whoever is doing good, like and people come to science in passion, right? People really like to do something. So I think, and I'm 100% sure that you're also passionate about science too. That's why you're in science. So as a chemist, like you're very well appreciated, like in biology, because as I said, like all the four biomolecule, like if we need to understand, like if, chemistry person will be better understanding the structure, right? How they are formed, and how they're like having the structures, and how, like saying that, like um, we like drug design, specifically drug design, and protein purification, like DNA purification, like all purification is based on chemistry basically. So like if we look for any job, I'm just talking about, like career perspective, that they prefer, like a chemist background mostly. So if you're in chemistry, like still I think you are connected to biology, like if you look at like protein or carbohydrate or something like that, any kind of structures. Ultimately, like people like, I'm just giving an example. My friend, like who is a chemist, like they do a lot of small molecule studies, like how they are formed, how they can be made. So, and those molecules we tested in our lab, right? So which are very well connected, I believe. And I think it's a very high demanding, like for chemists in biology. So if you want to pursue your research in biology, I think uh, as a learner, like you can get in, like any kind of any field of biology, and you can start from there. And I think it's a very easy, not easy, like very interesting subject. So we'll get interested, and we can solve a lot of problems.
Okay, sir. Thank you, sir.
Mhm.
Hello. Anybody have any questions? Anybody have any questions? You can ask directly to our resource person. Please unmute yourself and ask questions.
Some questions are seen in chat. You go chat and see questions are there.
Yes.
Okay.
Sir, Dr. Shoika, some questions are put in the chat box. You can have a look on them.
Okay, okay. So let me check. Yeah, my
So there is, I have not found any specific question in the chat box.
okay.
So is
Please give suggestion regarding PG. Okay. Yeah, I let me go through it. Like, yeah, I don't see that kind of specific questions.
hello.
Yeah, I can see, like, there's a I think student MSc Biophysics B21.
Yeah.
He's a student of MSc Biophysics. Please give me some suggestion regarding PhD in the near future. Yeah, like there are a lot of biophysics department. Yeah.
Yeah. Speak on. Hello, Dr. Saikia. Can you hear me?
Yeah. Sure.
You can,
yeah, I can hear you.
You can continue. You can continue. So there is a
yeah, no, yeah, this thing B2.
Okay.
Yeah, I see that there's a question that he's a MSc Biophysics and want to do PhD in biophysics. So to answer that question, there are a lot of departments, like in different institute. So I can tell that Indian Institute of Science Bangalore, they have a biophysics department itself. So they mostly do like bioinformatics, then protein modeling, protein drug interactions, and that kind of study. I know very well that we have collaborator when I was doing my PhD to study proteins and DNA, RNA. So I think if you really want to pursue like your PhD in biophysics, I think IISc Bangalore or IITs, they have a, I think, specific department for particular that biophysics. So you can do some kind of research on that. And maybe Guwahati IIT, I think they also have good department in biophysics.
So, Dr. Saikia, so
yeah,
I have seen some questions in the YouTube live. So I want to take one question from that. The Sweetie Rekha Gogoi asks, Sir, how do cells make accurate copies of DNA?
Okay. Yeah, that's a good question. See, we have a as I said, we have enzymes in our body, right? And if you look at the DNA is nothing. It's a nucleic acid, right? Deoxyribonucleic acid. So they are basically they actually bind to the protein and they form a stable structure called chromosome, and they are in the nucleus of the cell. So And when cell multiply, cell divide from one to another, it's a continuous process, right? Cell replicate basically, divide. So to do that, they have to be genome has to be duplicated basically. The copy of the DNA has to be duplicated, right? And to do that, basically they have to be relaxed from the protein. Then there's a particular enzyme called DNA polymerases in the cell. Then they bind to the DNA. Then with help of cellular resources like nucleotides, they synthesize basically the whole copy. So now your question is that exactly replicate, right? As I said, mutation thing. So they have to be perfect. There should not be any kind of error. Otherwise, whatever is duplicated, they will have a problem. They will not synthesize same protein, right? So saying that the DNA polymers have a proofreading activity, they can cleave off, if something is wrong, basically. If something is wrong in the synthesis, some wrong nucleotide is incorporated, so they have a property to basically remove that mismatch. So that's how they have the property, the enzyme, and they make the exact replica of the DNA. And now if you think about one step ahead, DNA is and DNA will be translated basically, not translated, transcribed to RNA, then RNA will be translated to protein. So now lot of time what happened, the DNA is perfect, but when the protein is synthesized, they are not properly folded. So there's a another step, in protein folding, basically if they are not properly folded, they will be basically cleaved and take it off from the cell. So there are a lot of kind of regulation inside the cell. It's very fascinating, how things happen. But to answer your question, with the help of DNA polymerases, they make exact copy of the DNA.
Hello.
Yeah.
There is another question from Amarjyoti Soikia to you. The question is, what are difficulties in vaccine management if virus itself is mutating in nature?
Yeah, that's a good thing. Very challenging. Basically, that is happening in case of influenza, that we have a cold, right? Common cold, a flu. So every year, they are mutating basically in their protein. Protein means surface glycoprotein. As I said, there has to be exposed in the surface of the virus. Then the influenza vaccine that we have, H1N1, that one, we every year, in India, we don't have that much problem with influenza, but in United States, every fall and winter, we have a lot of flu problem. So meaning that they mutate. So now how to overcome with that problem, right? Because they always mutate. But there's a what do you call that? There are particular branch of science that they do what they do. They always do a detailed study. Meaning, suppose the flu happened in this year, 2020. So they will study that what type of the virus is mainly there in the nature. Then they look back 2019 and 2018. So they go back basically how what particular strain is changing. So based on that, they make some prediction that this 2020 or 2021, they will have they will come like that, this type will come in next year. So they make a vaccine of that type, depending on the prediction. So that's how these influenza vaccines people are doing. So it's not 100%, but there's a chance that 70 to 80% is successful.
So, Dr. Soikia.
Yeah.
So I want to take one more questions from YouTube Live. So there is a question for you. That question is written in Assamese. So I would like to read it in Assamese. নমস্কাৰ ছাৰ, মই MSc কৰি আছো। বায়োফিজিক্সত MSc সম্পূৰ্ণ কৰাৰ পিছত কিদৰে আগবাঢ়িব লাগিব তাৰ বিষয়ে অলপ জানিব বিচাৰো।
Okay, very good question. Jodi biochemistry, তুমি biophysics কৰি আছা, no? You're doing biophysics, I think, MSc,
বায়োফিজিক্স
right? So there are two things. Like, you know, if you want to go to industry, like, you know, I don't know exactly what your career plan is, but you know, if you want to go to industry after completing your masters, better you do an MTech. You know, MTech. I know, like, you know, after doing MSc, like, you know, doing MTech sometimes doesn't make sense. But if you go to MTech program, 2 years program, you will have more exposure, basically, like, you know, to the application. Like in MSc, we have some limitation that we don't do that much technical. Like, we do more theoretical part. So if we go to MTech program, like in IIT or many other university have MTech biophysics. So if you go there, you will have a good training on, like, you know, how really you can apply your biophysics knowledge in a kind of a in technology transformation. Like, as I said, like, you know, protein drug design, X-ray crystallography, uh, that kind of study. So if you want to do if you want to be in the academics, I think I suggest you to go for a PhD program, like in a MSc PhD biophysics program. So where you'll have a, like, you know, extended training on biophysics. So where you'll have a like, you know, a particular project you'll be completing. Then after completing your PhD in biophysics, you can still, like, you know, join a company, or you can go abroad for your not going abroad, but you can do it in India as well, like, you know, your own lab, or you know, you can set up your own stuff, or you can have your own questions. Then you can set up your own lab and you can do your research. So I think both way you can think about it, like, you know, what's your career plan is, industry or academics, you can decide.
Hello.
Yeah.
So I want to take one more question from the chat box. The question is from Mayuri Soikia. The question is that there are various viruses in the world. Suddenly, another virus, COVID-19, arisen. What is the reason behind it? Is it formed by mutation? Please briefly explain.
Oh yeah. That's a nice question.
Jonale?
Because see, it's a very common question, like, you know, how this COVID-19 came, right? So if we say, like, you know, it's a brand new virus, it's not, because, like, you know, it's in the nature, it is there, and still many more to come. There are a lot of virus that still is there in the nature, and they are not infecting human. Currently they are infecting kind of chimpanzee, gorilla, or, like, you know, other animals. But what happened, like, you know, any virus, like, you know, have to be a pathogen of human. They have to do adaptation. Adaptation is nothing, like, you know, they keep on infecting. Like, you know, infecting means, like, you know, they're try to infect human. So over time, like, you know, they adapted to get infected. Like, you know, they can get they get entry, basically, to the human body. So this also happened like that. It was in the nature, but it was not infecting human. But I think because of some natural selection and adaptation, they came to human, and doing that disease, basically COVID-19, that whatever we are having it now. So um, so this is one example. Like, you know, you maybe heard about Ebola virus, right? So it was not infecting, but, you know, it was having in gorilla or chimpanzee, right? So people start eating raw stuff, like, you know, then you know, they had virus, like, you know, as a virologist, what I see is that, you know, they easily adapt to a particular condition. So they're parasite kind of stuff, right? So if we keep on giving it to a particular organism, like, you know, they start infecting. So that's how it came. And I don't know to scare you, but, you know, there will be a lot of virus come in coming years. Then we have to be ready, like, you know, that's why, like, you know, there's a demand of biophysicists that we can easily design the protein of the virus, which can be a good candidate for vaccine, and that can be like immediately used, right? So did I answer your question? I think that's how I can tell you, like as a virologist, because it is there in the nature. And I would like to add one point is that in a pre like before human, there were viruses, right, in the nature, and then ice age came. There then in the ice age, the virus flows in the glaciers, in a glacier, right? So now if like glaciers start melting, because of the global warming, so those viruses will be open to the nature basically. And human don't have any clue what they do. These viruses, they're maybe very dangerous, and we don't have any clue that how they will react, if they come to the nature. Now they are in the glacier, in the frozen state. And if it come to the nature, people has to be get ready how to tackle. So as I said, we do some kind of prediction, right? We can do prediction for the protein, prediction for the virus there, see if any new viruses will come, they will be not something that we don't know anything. We know something, that how their genetic material will be, how they will behave. We will have some kind of idea, but we have to be ready. So uh, there will be a lot of new things will come in near future. But I think as a biophysicist or molecular biologist, we have to keep on doing good research, so that we can do something.
So, hello, there's an announcement for the to the participant. So you can put your questions. You can send mail your questions to him. I will forward his mail address. So I have seen so many questions in the chat box. It is not possible to answer all of them. So with one more questions, actually, I want to close this question our answer session. So there is a one questions from Karisma Talukdar. Can you hear me, Doctor?
Yeah, I can hear you.
Okay, okay. There is a question from Karisma Talukdar. She asks, "Sir, I have passed MSc in Biophysics this year. I have interested on crystallography. I want to do PhD. So please give me some suggestions. How can I prepare for this?" So with these questions, we are going to close this question hour session.
Okay. So you're saying that you're a MSc biophysicist, right? And you're interested in crystallography, and you want to do research. So how to proceed, or how to make yourself prepared, right? So first of all, I would like to congratulate, because you're very clear with your vision, what you want to do. That's very good, because many people, many scientists, they don't know what exactly they want to do, because they are not very clear in their vision, but as you're what to do. That's a first thing, you're 50% already ahead, what you have to do. Now coming back to main point is that you have to do research in crystallography. So now if you Google crystallography research, if I'm saying India, if you put crystallography research comma India, then you'll see few scientists, in IIC Bangalore, or ICER Kolkata, there are few IIT Chennai, uh, and there's a scientist called M Vijayan, they're they're pioneer in biophysics in India. And you have heard about Ramasandran, GN Ramasandran, and who Ramasandran plot, he get it right. So he was in IIC, and I'm lucky to be in the same institute where I completed my PhD, GN Ramasandran. So Ramasandran plot, and without Ramasandran plot, you don't understand that much protein, right? How protein structure form. So saying that, you look for a good laboratory, in India, if you're going to do in India, crystallography lab. Then you'll see what they're really doing it, although they are saying crystallography, what they do, crystallography alone will not help you, because what is the application of crystallography, right? So you see that what kind of protein they do, and really what kind of downstream process they do, after protein is structure is solved, what they do. So then you see that how well it matches. Like after doing PhD, what you're going to do. You cannot do the same research your the supervisor is doing. So you have to look like 10 years ahead, like not 5 years, like your PhD time. After PhD, what you're going to do. So you have to have a long-term plan. Like I always say, like it's a kind of marathon, like not 100 meters. So you have to look for long term. Then now the problem will come. How will you get into the lab, right? First, you have to get into the institute, like or university. So you have to qualify the university exam or like institute exam, right? Or NET or GATE, right? Some institute don't take GATE, so NET. So you prepare like that way, like how to get to the institute, and if you almost sure that you will get to the institute, then what is the lab, and in that lab what they're doing, and try to like and understand what kind of research they are doing. So if you have these three things clear in your mind, I think you're almost 80% done. So rest is like your luck, like so I think that's how you prepare. If I am you, like I'll do that little bit of initial research, and I think that's how we'll go. And you have to do a little bit hard work too to get there. Is that answer your question?
Hello.
Yeah.
Okay. So we just want to close this question answer session. So do you want to speak more, Dr. Saikia?
No, I would
No.
like to conclude too. Like
Okay. 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 Okay.
yeah, two or three
Okay.
sentences that yeah, it's a kind of very interesting, and like I really like like the kind of session that what we have today. And I would like to thank Dr. Uza and Gyanjyoti and other faculty of Soidwar College for this very interesting session. And as I said in the beginning, that now everything become virtual. So we have to have a like social distancing and our safety things. So this is a very good way to communicate with a lot of people. Now we can see, like maybe we're 100 people here at the same time, right? So I think it's a good way to communicate. And second thing is that technology is advancing day by day, and a lot of information in the internet. We have to use it properly. So now, like today the topic was like biophysics and medicine. As you can see, like it's a just a part of in science, like biophysics and medicine. There are so many things to like understand. So as I said, like microbacterium tuberculosis, dengue, HIV, like there are a lot of things are there to solve. And the funny part, like I should say that there's a problem. That's why we have a job. Like we're doing something, right? So but it really need a passion to do science. It's not very easy. I will not say it's very hard, but you need a very dedicated uh, passion to do. So I encourage all of you to come to science and do something good, because whatever you'll do in science, it will be contributed to the people in our country or in the whole world. So, and all of you can like it is important, like all of our contribution. So with that, like I'd like to conclude. And Gyanjyoti, if you can, like give my email address to the participant, and if they have some specific question
Okay. Okay.
or they need some kind of any kind of advice, if it will be helpful, I'll be happy to do that.
Okay.
Thank you so much.
Thank you, Dr. Paramananda Saikia. I I hope you have addressed all the queries of our students very well. Not all the queries, maximum queries. So I will forward your email. They can put their questions. Now I would like to invite our one of the senior faculty, Homitto Barua, to deliver vote of thanks. Over to you, Homitto
Hello?
Barua.
I'm well. Am I audible to you?
Yes, you are audible.
Okay.
Okay. Good morning and welcome to all of you. Actually, it is good night, I think, for Dr. Soikia. And well, on the part of offering vote of thanks, at first, I would like to offer my sincere thanks and gratitude to Dr. A.K. Oza, Principal Soidwal College, for inaugurating this seminar. Indeed, we feel inaugurating effort. He has presented a relevance of this topic, biophysics and medicine, in a very dignified manner. We must take the opportunity to offer our sincere thanks, and we are privileged enough having Dr. P. Soikia, pride of Assam, presently working as senior scientist in USA, and having a broad and international reputation. We do offer him our sincere thanks for giving his valuable time and also making us all abreast in the said area, through which we are able to know how biophysics works in the new field of science and also how biophysics works in the field of like genomics and medicines. We feel it would have been better had we get him for a longer span of time, but unfortunately, time does not allow us. I think it is approximately midnight in USA. So very, very thanks, Dr. Soikia. We hope we will meet you again.